Thank you all. Thank you, Jeff, and thanks for the foundation for the invitation for the give us opportunity to talk about the our research at the Children's Hospital of Philadelphia. So my name is Dong Li, and I am a research scientist at the Center for Applied Genomics, and so. On behalf of Dr. Harknessson, let me give you a brief introduction about the Center for Applied Genomics. Dr. Harknessson founded the center in June 2006, and now we have a staff of over 70 staffs. And we have over 100 active projects with the Penn and the CHOP campus. And we did a lot of genotyping on the children's and also the adult population. So CAG informed consent form will also allow to recontact the patient. If we found something in the patient, we can always to recontact the patient to follow up if we need anything else. And then CAG database have intensively electronic health records, and as you know, it's unlimited. And we have intensive information on each child. And each year, on average, there's a 1.2 million visits to CHOP. And we have automatic robots to handling the samples. So here, just to give you an overview about our patient database. So CAG database presents the major disease area, including pediatric and adult disease. As you can see the breakdown over there, but I do want to emphasize that we also have over 22,000 rare disease samples. We have established over 100 collaborations over worldwide on the disease discovery and replication studies. So speaking of the sequencing project, we have sequenced over 7,000 whole exome sequencing on the rare disease, including epilepsy and other many Mendelian disorders. And we also have whole genome sequencing in the pipeline, and we sequenced like 900 typical neurodevelopmental kids, and 100 type 1 diabetes. And we in the pipeline we also have over 10,000 to be sequenced. And here just to show you our discovery and the development pipeline, we so combining family-based analysis, genomic database, and health records and phenotype information, we aim to identify biomarkers that underlying the diseases, and to try to come up a new therapeutic development, and then go back to the patients. We have done a lot, as I mentioned before, we have done a lot of whole exome sequencing on different type of diseases. As you can see, we are keeping doing this, and there's still a lot in the pipeline. Here, I just want to give you an overview about our previous work on 340 patients. Then overall, we have 40% diagnosis yield. So when we talk about diagnosis yield, what's the diagnosis yield? Diagnosis yield means you assume there's a mutation to be found in the patient, and you found the mutation, and you declare the mutation is pathogenic for the phenotype. But what is the next? You have a genetic diagnosis, and what's the next? So quite often, as you know, sequencing often leads to therapeutic dead end. As you know, there are over a few thousand genes that are responsible for rare disease disorders, and you may ask how many are treatable, and the answer is very limited percentage. So, just here, this picture from Norway, which is very beautiful. But if you take another angle to look at this scenario, you may find there might be a road, there might be a next to try to come up something to treat the patient. And here I just want to give you an example about our recent work on lymphatic disorders to illustrate how the genetic and genomic research can actually inform the therapeutic development. So here, just to show you the lymphatic system, just try to remind you this is a lymphatic system is a unidirectional drainage system, which are important to maintain the body fluid circulation and define the body against disease, and also absorb fats in the small intestinal. This is a this is a life-saving study. 2 kids present in the CHOP lymphatic center has fluid in the chest, and quite often stay in the ICU for several months. So here just show you how the lymphatic was disorganized and dilated in the different region, and there's a backflow to the chest. So I mean, the CHOP clinical team doesn't know what it's called. It's just have a name, lymphatic anomaly. So we did the sequencing as quickly as we can, and we found the same exactly mutation in ARAF in 2 patients, and this ARAF is very conserved across the different species, and is also important in this MAP kinase pathway. I'm not sure if I should talk about this, but when this mutation occurred, it turned on the whole pathway. It's just get the MAP kinase pathway stuck on a position, which means it will induce a lot of downstream things, and then lead to the phenotype. So when we have this diagnosis, we ask what's the next, and then we did the several studies in cells and also in the in vivo models. So we first introduced the mutation into 293T cells, and you can here just show the mutation will affect the interaction with the other protein, which eventually lead to the elevated MAP kinase pathway. And also we introduced the mutation to another cell type, which shows the similar, exactly same pattern, and another cell type. And again, we also tried a panel of FDA-approved drugs to see if they would be able to rescue the phenotype, by lowering the phosphorylation of ERK. And here you can see we test those 3 drugs we tagged can rescue the phenotype that induced by the mutation. And then we also test in the primary lymphatic endothelial cells, which shows exactly same conclusion. And then we also model mutation to see if mutation will affect the cell shape change. As you can see, when we introduce the mutation, the cells get disorganized. They get not round, they cannot attach to each other. And then when we gave the mutation the drug, Merck inhibitor, it can partially rescue the phenotype. Then we move forward to another cell type. As you can see, the mutation actually induce the elongation of the cell. The cell can be more thin, but and then they cannot attach to each other. But by the Merck inhibitor, it can reverse the phenotype. And here just the quantitative measurements for those phenotypes. We took a step further and also look at the sprouting activity, and to find out actually the mutation will spontaneously induce the sprouting in the endothelial cells, without the VEGF-C, as shown here. And again, the Merck inhibitor can rescue the phenotype. So here's the wild type, and then here's the mutant. At 30 nanomolar and also 100 nanomolar can perfectly reverse the phenotype. And again, those are just quantification, quantitative measurements. We also model the mutation in the in vivo zebrafish model. And we can see the mutation lead to dilated lymphatics, which mimic the phenotype that we observed in the patient in a few different lymphatics. And then we ask if the Merck inhibitor that we use in the cellular model can actually rescue the phenotype in the zebrafish. So as shown here, you can see that those. So in the wild type, the two Y cells can perfectly separate it, and you can tell there's one Y cell and there's another one. But in the mutant, you can see they are fused together. You know it's. You cannot easily tell which one is which. And after the treatment, the two vessels are separated. So here just again the quantitative measurement for the rescue study. And then we ask if we can use these results to inform the treatment. So before we identify the mutation, the patient tried the mTOR inhibitor, which is a standard practice for such group of patients. But the patient, before we identify the mutation, the patient has tried the Merck inhibitor, but didn't result in any significant improvement. So after we have these results in the cellular and in the fish, we switch the Merck inhibitor for this patient, and you can see the pulmonary function has been improved after 12 months treatment, and also so this is a before treatment, this is a 12 months treatment. Again, this is a before treatment, and this is after 12 months treatment. You can see there's intensively lymphatic remodeling, and there's a more normal appealing lymphatics in the patients. And also in terms of lymphedema in his lower leg, you can see there's a big difference in terms of lymphedema. So I think this is present a bedside to bench, back to bedside work. We start with the patient, and by accessing the sequencing technology, we can find the mutation, and then we model mutation in different cell types and also in the in vivo system, and then we successfully identified a drug that can be useful and to rescue the phenotype. Then we use that drug to the patient, and actually right now the patient resumed the daily activity. But before that, he was hospitalized quite often. So just summary, we identified the rare mutation in ARF that's responsible for this phenotype, and we modeled the mutation in different cell lines and confirmed it lead to enhanced MAC activity. And then transgenic study in zebrafish also confirmed the mutation is a gain of function and actually mimic the phenotype that we observed in the patients. And then we successfully identified a drug that also successfully applied back to the patients. So let's talk about the Champ1. If we could do the similar thing or different or similar concept to try to come up with something that might be useful for Champ1 patients. So first, Champ1 genes has 3 exons, and so has 2 non-coding exon and 1 coding exon. As you can see, the poly2 and the other histone markers shows the transcriptional start site in the first non-coding exon, and as you may know, all the mutations identified so far in the 3rd coding exon, and you may also heard something like nonsense immediate decay, but but this nonsense decay will require the 50 base pair rules, which means a stop gain or truncating mutation has to be located before the last 50 or 50 to 55 base pair of the second last exon to introduce the nonsense decay. But all the mutation, like I said, in the last exon, which means this will escape the nonsense decay. Indeed, we did RNA-seq on JJ, and we found the mutation actually has a relatively comparable level to the wild type. We found the 12 rays has this mutant allele, and the 17 rays has a wild type allele. In fact, in 2016, Coury's group from France has looked at the mutant protein, and you can see all those mutant actually express the short or truncated protein, which means this Champ1 mutation escape nonsense immediate decay. So in the cells, the truncated allele is still there, but we don't know if the truncated allele still have some function or not. But let's look back to the 2010 study by I think by Dr. Tanaka from Japan. He did a few constructs by deleting several domains of this gene, and basically shows the last three link finger domain are important for this chromosome and this spinal localization, as shown here. Those positives here, but he also showed this FPE motif is also has some function in terms of the spindle localization, as shown here and here. But in terms of other motif or domain, we don't know exactly what they do. But as I said, the mutation allele is still in the cells. So here just show how this truncating allele looks like. It will have, some may just lost the three ring finger domain at the C terminal, and the others will have a shorter isoforms. And also in 2017, there's a study showed the WK motif has interaction with another protein, also very important in the mitosis process. It's called MAD2L2. And after this interaction, they may form a dimer structure, to be function. So this is this raises another question. If this truncating protein has some function, dominant negative or gain of function. So I know you from literature you see a loss function effect because it's truncated the last three ring finger protein. But if the truncated protein has some truncating protein is still there, they have some motif. They may band other different factors, during the mitosis. So it can have some different effect. So that's what we are trying to figure out. And so that's why we come up with this plan to do the IPF cells. No, so there's a, you can think about this as a complex to be functional, and if one of them has some defect, it's still there, and the other part of the protein, can band to other have an interaction with the other factor. It's not the whole pathway get defect. And there's a study I think from 2016 by Japan group. They look at the patient cells. There's only like 14% of the patient cells had the spinal defect. You can imagine, this is a heterozygous mutation, and there might be 50% defect, but the study only observed like 14%. There might be some, either the cells are introduce the cell death process to to protect the effect, or there's some other mechanism at play. We have to figure out. Yeah.
A lot of the literature talks about a complete loss of function.
Yeah.
Are you saying that there's a chance that there's still some function with the truncated or with the shorter protein?
Basically, yes, but we are not sure.
Or how it affects?
Yeah, how it affects. There's other. So as I shown here, there's different motifs. You can see this. They show the FPE motif has some function in terms of the spinal localization, but we don't know, if there's the N-terminal link finger protein domain also has something, they can bind to some other factors to be function. We just don't know, how complex this complex is.
So do we know what the other aspects of it does, I guess?
No,
we know what the last three do, right? I mean, that's what establishes a lot of the things that our children suffer from, but do we know any of these other things, how they affect the children?
We don't know.
We don't know yet.
Yeah.
Okay.
Yeah.
Thanks.
Yeah. Okay. I'm almost done. So our future plan will to generate the iPS cells. So as you know, we can start with the blood or skin biopsy to reprogram the cells to iPS cells, and basically after that we can branch out to any cell types that we desired, and use that model. We can look more closely at the pathogenic, how this mutation affect the cell development and how this lead to the diseases, and we can test the drug, and we can test the drug toxicity. And then, after that, we may, there might be a chance we can use that to give to patient, like the example that I gave. So there's a few advantages about the iPS cells. It's human origin. It has the same exactly genetic background as the patient. It's easy accessible and easy to give rise to almost any cell type we are looking for. And those cells are actually immature and often more similar to fetal than adult cells, which is make this perfect model to model this early onset disorders. And then the iPS cells has a advantage in the precision medicines to incorporate CRISPR, to correct the mutation and see, if the iPS cells line will behave similar to wild type, and identify the new compound and testing the drug. With that, I would like to thank Dr. Harkness and the other colleagues in CHOP, and especially to Jeff and Kat and the Champ1 Foundation and patients. Thank you very much.